Let's talk about enzyme-coupled receptors. So remember that these are receptors that either are part enzyme themselves or have a domain that is an enzyme, or they are associated with another enzyme. So let's look at a more specific example, and that is receptor tyrosine kinases, or RTKs. Um, so this one is specific in that it acts on tyrosine amino acids and phosphorylates their hydroxyl groups. Now, we talked about protein kinase A and protein kinase C in previous videos, and those are examples of serine or threonine kinases. So what we have is these two parts of the RTK, and then once you have a signal molecule, it'll bring them together or dimerize them. And then, since they're so close together, the tyrosine kinase domain, which is the enzyme part, is going to interact with the other tail and phosphorylate it because kinase adds a phosphate. So it's adding phosphates to this one over here, and this tail is also adding phosphates to the other tail. And now all these phosphorylated tyrosine amino acids become binding sites for other signaling molecules or proteins to come and bind to them and continue the signal pathway further. One of the proteins that can attach to the phosphorylated tyrosines on this RTK can be adapter proteins. And these adapter proteins can help activate these RAS proteins. And RAS, they're a type of G protein, except they're not like the ones that we saw previously with the whole GPCRs, because those ones are trimeric. But these ones are monomeric, so just one part. But like the alpha subunit of the other G protein, these also have an intrinsic GTPase activity, which means that they're activated once they have a GTP bound to them. But then once that happens, they have this clock, and it's kind of like a timer, and when it runs out, they hydrolyze the GTP, so they take off the 1-phosphate and it becomes GDP, and then it's inactive again. So these adapter proteins activate it, so it makes the GDP dissociate from the RAS, and then GTP comes and binds to it. Now you have an active RAS protein, and RAS is important in cell growth, differentiation, and survival. So if we have a problem in the RAS protein and that it is no longer able to turn off the GTP, its activity, so it's permanently activated, even when there is no signal coming in. That can be really bad because it has uncontrolled cell growth and differentiation and survival, which often leads to cancer.